then the cytoarchitecture, neural circuitry, and the clinical importance of the cerebellum. Alright. Now, as you know, the cerebellum is in the posterior cranial fossa covered by the tentorium cerebelli, and in the midline of tentorium cerebelli, this is a posterior cranial fossa covered by the tentorium cerebelli, and in the midline there is falx cerebri is attaching. Falx cerebri is attaching, and within this falx cerebri there are two sinuses, that is the straight sinus, and along its posterior border there is the transverse sinus. Got it? And from it towards the occipital sinus there is falx cerebelli with the occipital sinus. Cerebellum has a superior surface and inferior surface. Anatomically, it is divided into anterior lobe, medial lobe, and the posterior lobe. Superior surface, inferior surface, right? And embryologically, it is divided into archicerebellum, paleocerebellum. Archicerebellum that appears in the aquatic animals. Paleocerebellum that appears in the terrestrial animals with the development of the trunk, or limb muscle, with the development of the limb muscle, right? And the neocerebellum with the appearance of dentate nucleus, it is in the primates and mammals, including the human being, that helps us to perform skillful, accurate and skillful actions, right? So, archicerebellum is mainly vestibular in function, that maintains the equilibrium and posture of our body. Paleocerebellum maintains the tone of the muscle tone and the tone of the trunk muscle, and divides the body into superior part and inferior part. And in case of the neocerebellum, it receives input from pontocerebellar fibers, inferior olivary nucleus, and fibers from the exteroceptors and proprioceptive fibers. Alright, that's why it can perform and help in the accurate prehensile work that we do in drawing, in writing, in painting, in for the musicians, alright, all the, and dancing, I must mention, and in dancing it helps the neocerebellum, right. Let me draw the picture of the gross anatomy of cerebellum. divided into the midline vermis, worm-like vermis, and on the lateral side there are the cerebellar cortex, just like the gyri and sulci of the cerebral cortex, right? Only 50% of the cerebral cortex is exposed to the surface, 85% is deep into the sulci. Now, if we subdivide the vermian part, we will see there are lingula, central lobe, culmen, decleve, folium, tuber, pyramid, uvula, and the nodule. Actually, the nodule and tonsil comes in its inferior surface together with the uvula and pyramid. And the lateral extension of this vermal part, we see that there is ala, quadrangular lobule, lobular simplex, then the superior semilunar, inferior semilunar cortex, then comes the biventricular cortex. Up to this, we constitute the superior surface. Below that, we see the inferior surface, where there are two important structures, that is the tonsils. Tonsils are circumscribed cerebellar substance, which are connected to the uvula by a white matter band called the furrowed band. You may remember or not. The tonsil is below a white membrane that is known as the inferior medullary velum. You have seen the superior medullary velum at the junction of the two superior cerebellar peduncle. I gave you last day, there is superior medullary velum at the superior angle of the fourth ventricle. Alright, and from there comes out the trochlear nerve. We are seeing this superior medullary velum. So, above the tonsil, not below the tonsil, above the tonsil, there is inferior medullary velum. Alright, that is a white membrane under which it lies. There are fissures, that is the fissura prima, in between the culmen and decleve. There is horizontal fissure in between the inferior and superior semilunar cortex, and below that you see the lateral border is extended up to the postlateral fissure that comes behind the flocculonodular lobe. You can see, but in this picture the posterolateral border comes above the flocculonodular lobe, but I am telling that it is behind the flocculonodular lobe. How? Because vermis is not like this, straight or perpendicular, vertical. So it is carved like this. So if this is nodule, then the flocculonodular, the posterolateral sulcus will be behind it, not in front. What my point? As it is bent like this, worm-like bending, so the floccular nodule is here and the posterolateral border is here. That is behind the nodule, and the nodule constitutes a vermal part, and laterally with the peduncle a flocculus is attached, which is vestibular in function. Right? Now, I have described it paleontologically. There is also a longitudinal subdivision. That is median vermal portion, medial, lateral to the medial part, that is the paravermal portion, and lateral zone, the lateral portion. The median portion is projected to the nucleus fastigius, and it receives fibers from the spinocerebellar and vestibulocerebellar pathway. Then comes the paramedial portion, which projects to the nucleus interpositor, that is nucleus globosum and the nucleus emboliformis, and it is projected to the nucleus globosum and the nucleus emboliformis, and it receives the inputs from the rubrospinal tract. Either first one was vestibulospinal and reticulospinal. This is rubrospinal tract. Then the dentate nucleus that receives fibers from the inferior olivary nucleus and projects via dentato-rubro-thalamocortical fibers, corticospinal or pyramidal fibers, and the rubrospinal fibers. Got it? It is easy, not that much complicated. But the complicated part is the internal architecture of the cerebellum, which is better given than any anatomy book in the W.F. Ganong. If you read the Ganong, the description of the internal architecture is given in a very good fashion. You will understand better than any other, whether AKT, BDC, Samar Mitra, Vishram Singh, all are poor in this region, but W.F. Ganong is very good if you try to understand the internal circuit. What are there? There are 3 layers. In the deep core, there are the deeper nucleus. Which I didn't do. From medial to lateral, there are nucleus fastigius, nucleus globosum, nucleus emboliformis, and dentate nucleus. All right. In the midline, there is nucleus fastigius. Paramedial is the globosum and emboliformis, and lateral is the dentate nucleus. Okay, if I get the time, I will draw the picture. And the cerebellar cortex has 3 layers. It has the molecular layer, intermediate layer of Purkinje cells, and innermost layer of granular cells. Molecular layer, intermediate layer of Purkinje cells, and the granular layer, innermost layer of granular cells. All right. In the molecular layer, what we see, there are the parallel fibers from the granular cells, and these are axons, not dendrites, and dendrites of Purkinje cells. They are entering into the molecular cells, and dendritic cells, the dendrites of the Purkinje cells makes spinous dendrites as well as non-spinous dendrites, which are spring-like, and the mossy fiber comes to encircle this non-spinous part of the spiral pyramidal tract, right? In addition, there are stellate and basket cells also in the molecular layer. In the Purkinje layer, we see the cell bodies of the Purkinje cell in an uniform status, whose dendrites enters into the molecular layer, and the axons go down into the deeper layer, which provides synaptic connection to the deeper nuclei. And the inner granular layer there are granular cells. These granular cells, the action terminals forms a rounded swelling which forms the rosettes, which is the rosettes is the terminal part of the granular cell which is dilated, that forms the central structure of the cerebellar glomeruli. If you are asked when you are given a kidney histology of kidney, you will be asked, have you ever seen glomerulus elsewhere? You must be able to answer, yes sir, I know there are cerebellar glomeruli. There are cerebellar glomeruli. In which layer? In the granular layer. From the granular layer, the granule cell comes out, forms a dilatation, and it is covered by actions of the Golgi cells and the dendrites of the granular cell, together with some glial cells which are known as Bargman glial cells, Let me draw a rosette and glomeruli. As you see, from medial to lateral, within the deep core of white matter, there is nucleus. Medial to lateral, there is nucleus fastigious, nucleus globosus, nucleus emboliformis, and the dentate nucleus. Below, I have drawn the medulla, and there is the fourth ventricle. Did you understand? Now, let me draw a rosette. How a rosette forms? after drawing a diagram, you should always write the anterior, posterior, lateral, medial together with a caption, that is the figure shows superior surface of cerebellum, figure shows inferior surface of cerebellum, figure shows cerebellar glomeruli, like this. Alright. Now you can see the end of the mossy fiber is dilated, and which is surrounded by the actions of the Golgi cells and the claw-like dendrites of the granular cells, and the gaps are filled by the Bergman glial cells. This constitutes the cerebellar glomeruli. Understood? The terminal dilatation of the mossy fiber together with the actions of Golgi cells and the claw-like expansion of the granular cells, and the gaps are filled by the, like the kidney, there are some podocytes, here there are some Bergman glial cells. This constitutes the cerebellar glomeruli. Up to this much, I think there is no confusion. Alright. Now tell me the summary of, you don't try to write the entire circuitry of cerebellum. You just try to write only the summary of internal circuitry of the cerebellum. You will say that out of the 5 type of cells within the cerebellum, that is the pyramidal cells, Golgi, granular, stellate and basket cells, only granular cells gives the positive input to the deeper cerebellar nuclei, and all others are produces negative impact, gives rise to the negative influence, right? And finally, when the fibers come out as the output from the deep cerebellar nuclei, this is also negative in nature. This is also negative in nature. So the out of the 5 cells, that is the stellate, basket, granular, Golgi and Purkinje cells, only granular cells have the positive influence on the deeper nuclei or other cells, and Golgi cells is particularly have the negative influence, and all other cells are also have negative influence. Now what happened when the and the input to the deeper nuclei, that is the mossy fibers and the climbing fiber, both produce positive influence to the deeper nuclei, the mossy fibers and the climbing fibers. The mossy fibers come from the pontine nucleus and spinal nucleus. You should remember this. Mossy fibers come from the spinal nucleus and pontine fiber, and parallel fibers may origin from the brainstem nuclei and other nuclei of the pons or medulla. Now when the Purkinje fibers exerts its negative effect, together with it influences the negativity of the other cells except the granular cells. So this is called feed-forward inhibition. This is called feed-forward inhibition. It is inhibiting the deep nuclear nucleus as well as it is inhibiting other negative influence to the deep nucleus, that is deeper nucleus, that is the granule cells, except granule cells, Golgi cells, stellate cells, basket cells, right? So these will be facilitated and will produce more inhibition to the deeper nuclei. So this is called feed-forward inhibition. Another thing is that when the negative influence comes to the deeper nuclei, the granular cell, it stimulates the other negative cells, but it itself produces positive effect towards the deeper nuclei. So in deeper nuclei, and reduces the inhibitory effect of other cells. This is called feed forward inhibition, that is done by the mainly by the granular cells. So finally, he will get negative inputs from the deeper cerebellar nuclei. Right? This much is enough. Clinical importance, as you know, the static tremor, incoordination in movement, cerebellar ataxia, dysdocokinesia, the increased muscle tone, and incoordination. That is, if you ask the patient to touch the tip of the nose, his hand will go this side, it may go above, it may go down. If you give a small gulp of food in his hand, he will not be able to put it in his mouth. It will go toward this side, it will go toward this side, this side, this side. This is known as pass pointing. And cerebellar injury also produces trunk muscle weakness, and there is ataxia, that is called drunken ataxia. The person gait will be like a drunken person. Alright. There are many others, I will tell you later on. Already. Let me finish. Let me conclude up to this. If anything remains, I will tell you later on in course of discussion. Okay. Thank you very much for listening to me patiently.